All right. So first, we are incomplete now. It's nice. It's nice. Welcome back. Any questions? Any questions so far? So we analyzed until the figure 4. We finished the figure 4. So if there is any question on the method or on the meaning of the discoveries or whatever, any question is fine. So we finished figure 1, 2, 3, 4. So it's kind of interesting that there were papers in the 1970s that reported that messenger RNA had M6A. But because of the technical difficulties at that time, many scientists thought that actually the M6A that had been found originated from contamination from ribosomal RNA and tRNA. Because ribosomal RNA and tRNA, it's about 98% of the total RNA amount inside the cell. So scientists tried to purify messenger RNA and then to cut messenger RNA into single nucleotides. And then they showed that they were methylated adenosine. But then other scientists said, well, you have not purified messenger RNA completely. In your mix, you are still having ribosomes and tRNA. So many people did not believe that messenger RNA was methylated. And it's only in 2012 that 2 papers, one in Cell and one in Nature, conclusively showed that there was really M6A in RNA. And they did that by using RNA sequencing, by immunoprecipitating methylated RNA and then sequencing, clearly showing that the sequence of methylated RNA was messenger RNA sequence. So then this was that was the confirmation that really M6A was methylated. But the problem is that even though it was known that it became certain that RNA was methylated, what was the physiological function of RNA, of M6A, was not known. And even now, it's not known. Most of the papers, as this one as well, but the papers that are published this year as well, and the year before, it's all in vitro, only using cell cultures. And so far, physiological function of messenger RNA M6A methylation in adult animal has not been demonstrated. One of the big problems is that if you make knockout animal who lack the enzyme that writes M6A onto the messenger RNA, these animals die. They die before you get them because they die in utero. They don't develop. So the only thing that we know is that it's essential for development. And something that has been done in ES cells, in embryonic stem cells, is that RNA methylation is important for differentiation. If you don't have RNA methylation, the cell does not differentiate. This is the reason why the animals die, obviously. But this does not explain what is in you now and in our cells, in mice, what is the function of M6A for normal metabolism, for normal physiological process? We don't know. So this is why we decided to do this kind of things. Any question? No? No? Okay. All right. So then, please ask a question. I will ask you a question. So we have shown so far that when we use drug, diazoadenosine, to inhibit our methylation globally in the whole cell, we see that we affect RNA. And when we quantify M6A, we see that actually M6A of messenger RNA decreased. Okay? And the same drug that inhibits RNA methylation also affects the circadian period in mouse, in human cells, in mouse cells. But this is not enough. Our drug inhibits many other methylation, as it is shown in the supplementary figure, which I will show you later. Other methylations are affected, like histone methylation. What is the next step? What must we show? It's a paper on RNA methylation. So what must we show now? What's the next step after this one? This was only the confirmation that, yes, deazoxyribonucleic can inhibit M6A RNA methylation, but it does not inhibit the methylation of everything because cap methylation is not affected. And when we inhibit RNA methylation, M6A, then we expect to see differences in RNA processing, which is what we got. And when we check RNA processing means the export from the nucleus to the cytoplasm, it seems that is indeed affected. But what next? What's next? Can you sort of give some experiments? I'm not asking you to tell me what we did, which you may know already. Of course, you can tell me. But what would you do? What would you do? What would you do after these experiments?
Increase the RNA methylations whether there is an opposite effect.
Yes, you could do that. You could increase RNA methylation to see if the effect is opposite. And this is what we did at the end. So we will come back to that. Yeah, that's a good idea. But I have to say that at the end of figure 4, we cannot say the title is not good. If we only show up to figure 4, and if we say RNA methylation is important for the circadian clock, it's not good. Because the data so far does not show that RNA methylation is important. It shows that our drug, yes, inhibits RNA methylation, and it shows that our drug affects the circadian period, but it doesn't show that inhibiting RNA methylation elongates the circadian period. Maybe our drug inhibits the methylation of something else, and this affects the circadian clock. But RNA methylation doesn't matter. So there is no causality proof here. We have not demonstrated the causality between RNA methylation inhibition and the elongation of the circadian period. Do you understand causality? What it means? Do you understand causality?
Cause and effect relation.
So, what do we do?
We may modify RNA M6 and
What do you mean modify RNA M6? What do you mean modify?
Prevent methylation sort of cap and add the DAA and we may show period elongation doesn't happen.
So you mean you want to use DAA and?
Other than RNA methylation doesn't occur, doesn't change period.
So what is the experiment you want to do? Explain again.
Use some drug
To inhibit other methylation.
to protect the RNA M6 side.
Okay. You use DAA, but in addition you use a drug that sort of protects M6A. That would be a good idea. That would be a good idea, but there is no such drug. There is no such drug. So you may not know that, although I told you in the previous lecture, you weren't there, but all methylation reactions require S-adenosyl methionine. S-adenosyl methionine is used as a donor reaction, as a donor, the molecule that gives methyl. But the enzyme that is using SAP and methylates substrate, DNA, protein, fatty acid, RNA, cap, M6A, whatever, they are different. There is one methyltransferase enzyme for each substrate. There is a methyltransferase enzyme that methylates a specific nucleotide in ribosomal RNA. There is a specific RNA methylase that methylates a specific nucleotide in tRNA. There is RNA methylase that methylates the methylation of the cap, the M7G. Every single methylation has its own methylase. Sometimes there can be more than one. Sometimes there can be more than one, and some methylases work together. So you have 1 and 2, and they work together. So knowing that, what could we do?
Probably can apply while you apply DAA, in addition you use a very high dose of the enzyme that specifically mesotransferase that works on RNA. And if actually by doing this you can reverse the effect of DAA, then I think it somehow shows that it's the RNA methylation that explains.
That's a good idea. So if you make an excess of the RNA methyltransferase, and in the same time as treating your cells with DAA, you actually force your cells to make a lot of enzymes, then you may sort of decrease the effect of DAA. That's a good idea. That's a good idea. However, what is difficult is to produce enough proteins for that. But you could do. You could do. You could maybe transfect your cells with a DNA fragment that codes for the methyltransferase enzyme that makes M6A, and maybe these cells will be protected. That is possible.
You could look up the enzyme, only the enzyme which is responsible for that blockade.
Yeah, I like that. Knockout. ideally, if you have all the tools available, this is what you do. This is really the most powerful and direct method to investigate something specific, is to do the knockout. You remove from the animal the protein that methylates messenger RNA at the internal M6A position. However, as I said, at the time of this paper, there were two proteins known to make M6A. It was METTL3, methyltransferase-like 3, and methyltransferase-like 14. And they work as a dimer. But unfortunately, when you remove METTL3 from the animal, the METTL3 is the main one. But when you remove it from the animal, we tried, the animal died. So we cannot do knockout.
Knockdown?
Knockdown. That's what we did. Do you know what is the difference between knockout and knockdown? So knockdown is you are using RNA interference to decrease the expression of a given protein. RNA interference. You know RNA interference.
I forgot.
RNA interference is very simple, is that you imagine a messenger RNA, it's single-stranded molecule, and there is an antiviral mechanism in the cell that gets rid of strange RNA. If you have a strange RNA in the cell, there is a mechanism in the cell that will remove it. So what we can use this system, we can hijack this system, and we can trigger the degradation of specific RNA molecules in the cell. So what we simply need to do is to inject into the cell a small bit of RNA that is complementary to the target RNA you want to decrease. So we know the sequence of the messenger RNA coding for METTL3, so we just need to add small complementary RNA that will hybridize to the METTL3 messenger RNA, and then the protein METTL3, the expression will decrease. This is in vitro experiments. We don't do that in vivo normally. There is a way, but we don't do it normally. And this is the data here. So this is using mouse cells, I think. No, this is human cells, and this is mouse cells. And we can measure the biological clock by following rhythms of luminescence. It's the same cells as we used previously. And here you can see very nicely, so this is control-treated cells, and number 48, so the messenger RNA coding for METTL3 is quite long. So you can use small bit of RNA that are targeted to different sequence of the messenger RNA. Okay, it's very long. Ready?
Suddenly disappeared.
Suddenly disappeared. Yeah, I saw people frown. I was wondering. There you go. So we actually used two different small antisense RNA molecules, number 48 and number 49, and as you can see, they both efficiently decrease the expression of METTL3. This is a Western blot where we quantify the expression of a given protein. This band means that there is a lot of protein, and you can see the band here is much less intense. It means that the amount of protein is much lower. However, if we check actin, which should not decrease because this antisense RNA is supposedly specific to METTL3, so when we do that, it should not affect actin, and indeed, the actin is not different. And this is in human cells, and this is in mouse cells, and as you can see, it's the same thing. The METTL3 knockdown is also very efficient, and it has no effect on actin. But, as you can see, knocking down METTL3 is enough, is sufficient to cause the elongation of the circadian period. Especially, you can see number 48 is very powerful. The period almost goes to up to 30 hours. So only knocking down MTTL3 is enough to cause an elongation of the circadian period in human cells and in mouse cells. So we can recapitulate the effect of deazoxyribonucleic by simply decreasing the MTTL3 protein inside the cell. And then we decided to test different doses of the RNA antisense, because the more you add, the more your MTTL3 will decrease. So then we should expect to see a dose-dependent increase of the circadian period when we increase the amount of siRNA. And indeed, you can see we tried 2 different doses. This is in human cells. We tried the control siRNA at 10 picomole, or whatever, it's 100 picomole, and then 250 picomole, so it's 2.5 times higher. And we did the same thing with siRNA number 48. And as you can see, using 100 picomole of the siRNA number 48, we have elongation of the circadian period. But 250 picomole, it makes it even longer. So we have a dose-dependent increase of the circadian period. And then, very nicely, this period here, it's like 35 hours. So you have a circadian clock, which is supposed to give you 24, 25 hours in vitro. And without RNA methylation, you get up to 35 hours. So this is a period elongation of 10 hours, which is quite dramatic. And I don't need to explain this one, but next, this is your idea. We show that when we decrease MTTL3, the circadian period goes longer. So if we increase MTTL3, we should promote RNA methylation, so the period of the clock should get shorter. So this is exactly what we did. It's maybe let me make it bigger, so we can see. So this is exactly what we did here. And here again, it's protein analysis. And we are detecting MTTL3 here and actin as a control. Actin should not change. We are not doing anything with actin. But MTTL3 should change. So you can see the lower band here is the MTTL3 that is already in the cell, the endogenous MTTL3. What we are doing is that we are adding new MTTL3. But the new MTTL3 that we are adding is marked by a specific thing, which makes us able to detect this additional MTTL3. And you can see this additional MTTL3 is it's a bigger protein, a little bit bigger. It has the same structure, it has the same function, but it's slightly bigger. And you can see it's here, there. The band that is on top of the endogenous MTTL3 band, this is this MTTL3 MYC flag protein. So a little bit big. It is tagged by a different peptide. But you can see that in non-transfected cells, in control cells, you only have MTTL3 protein. But in cells that are forced to express this new protein, suddenly there is a new band appearing. This is a different type of cell that is also expressing the same thing. And again, we have a band here. And then there is delta and delta. So that's kind of interesting. What we did here, we found that there is a paper in yeast, because this M6A methylation also exists in yeast. And the sequence of the yeast homologue of MTTL3 is almost identical to humans. And what they did in the yeast paper, they actually make a mutant of MTTL3. And it's a mutant that has no activity. It's the same protein, but it does not methylate. So this is what we try to do as well. We try to express more MOTTL3 in the cell, but we also try to express a mutant version of the protein. So the amount of protein increases, but the activity will not increase because it's not effective. And then we use these cells to measure the circadian period. And what we saw, as you can see here, so in black, this is the cell that expresses. This one doesn't express much, but you can still see the band here. And this cell expresses a bit more. So these black and gray, they are the two cells that express the mutant version of the protein. And you can see they have a period that is 28, which is a bit higher than normal cells. However, the cell that express more of the wild type of the active MOTTL3, they have a shorter period than the cell that express a mutant form of the protein. So indeed, it seems that overexpressing a functional MOTTL3 causes a shorter period compared to expressing a non-active mutant protein. All right. So what's next? Do you have any question? It's not perfect. It's not perfect. Do you have any comment or question?
Why did the cells die when the genes completely locked out?
That's a good question. That's a good question, and there may be different reasons for that. I think the main one is simply that knocking down will not completely remove the protein. Some small amount of protein will remain that can maybe prevent the cells from dying. On the other hand, the cell that you have in your dish is not an animal who is developing. So a cell can get away by losing quite a lot of genes and still be fine. So that could just be the case. That in cell culture, even there is no MOTTL. Actually, I did, using CRISPR-Cas9, I did a completely deleted MOTTL3, and the cells can survive. It's just that during development, MOTTL3 is needed, so the animal cannot develop. But once the cells have developed, you can lose MOTTL3. You mean this one? Yes, that's true. So there is a difference here between this one MOTTL3 delta and two MOTTL delta. And it seems to be significant here, but there is no, still you can see here.
If there is something in the fourth sample, then we can explain that because we add the mutant MTTL3, then the cell will think that is enough and there will not be more MTTL3. There is no mutant MTTL3 in the fourth sample.
Yeah, there is. You can see a little band here. It's very faint, but you can get it. This one here. You can see? But it's not perfect. We would have liked, actually these, what are these cells is, what you do with cells, you can insert into the cells a plasmid. Do you know what is a plasmid? That will express the plasmid, it's a bit of DNA. You inject into these cells a bit of DNA that codes for MOTTL3. But in this case, in addition to that, we made single cell-derived colonies. These are monoclonal cells. These are monoclonal cells. We wanted to have monoclonal cell lineage. And unfortunately, they were all different. Some of them express a high amount of protein. Some of them express a lower amount. But you are right that, so the cell 1 here and the cell 2, they are completely different. They are different lineage, monoclonal lineage. But what we would expect to see is that maybe the number 1 here express more of the mutant MTTL3, so the period should be longer, right? But it's the opposite. So there is obviously something a bit strange there. And I think it's due to the fact that MTTL3 does not act alone. There are other proteins that are involved there, like MTTL14 is also involved in RNA methylation. Here we are only using one protein, so there may be something, and there are still things that are not explained now. So we don't explain 100% of what we see here, but in average, we can see that the cells that express the wild type, more of the wild type protein, have a shorter period than the cells that express a mutant version. And we can see very clearly here, the period is much shorter of the orange one compared to the any other questions or comments? No? Okay. So, what's next? What's next? Now we have shown that changing the levels of MTTL3 is enough to actually change the circadian period. So what else? Obviously, we are currently doing MTTL3 knockout mice, but I told you they are dying. So what could you do? We tried to remove MTTL3 from the mouse, but mouse cannot develop, so we stopped. But we are doing something else. When the gene that you are removing causes the mouse to die, what can you do?
Use an inhibitor once it's been developed.
Yes, you can use inhibitor. Unfortunately, there is no specific inhibitor of MTTL3. So what could you do? You still want to use, obviously, all in vitro work is interesting, but the impact of in vivo work is much better. It's much higher. So what we want to have is a mouse that does not have MTTL3. But they are dying. So what step forward can you make? How can you deal with this? And it's something that is very common to many research.
Mutate it. So that it still functions.
Yeah, you can mutate. We could have done the mutation there, but maybe we could have done in mouse the mutation that we did here. We could have done. Maybe the cells would have, the mouse would have died as well. I don't know. But it's something we could do. But we could potentially remove MTTL3, but in a specific way. In what manner of specific way? Specific to what? Like, what is the main location in the body or the main tissue that is the most important for the biological rhythm of the animal? CN, the suprachiasmatic nucleus.
Tissue-specific knockout.
Yeah, you could do a tissue-specific knockout. And this is what we are currently doing. So we are removing MTTL3 from the SCN, specifically in the SCN of the mouse. How can you do that? That's an excellent question. We are also doing this in the liver. Because we want to know not only if MOTTL3 is important for the circadian clock in vitro, but also whether it's important for metabolism. And the liver is a highly metabolic tissue. And we have detected many transcripts with high methylation level in the liver. So we are wondering what will be the animal, what will be the phenotype of the animal. But the question is, how can we do that? How?
LogSP system.
Cree LogSP system, yes, very good. What is Cree? What? So the Cree LogSP. Let's write this. I think that will take some time. This sounds difficult, but it's very easy. Cree LogSP system. Cree is the name of an enzyme, and LogSP is the name of a specific DNA sequence. Cree, as I told you, it's an enzyme. More specifically, it's a recombinase. It's a DNA recombinase. It means it's an enzyme that will change DNA. It will change DNA. And very simply what it is, if you imagine a sequence of DNA in which you have whatever the sequence, you have one LogSP, and a little bit further, you have another LogSP. The distance here can be anything. It can be anything. It can be short or it can be long. When the Cree recombinase, the enzymes, sees such a thing, so a fragment of DNA flanked by two LogSP sequences, what the enzyme will do is like this. It will remove whatever is between the two LogSP. And at the end, you have the same DNA sequence. The sequence before the LogSP is fine. The sequence after the LogSP is okay. But essentially, you have lost the entire sequence that was between your LogSP, and you're also losing LogSP. You are losing one of the LogSP sequence. I don't remember if it's the one in the front or the one at the back. I don't remember, but you are using one LogSP. So, how can this be used to make a tissue-specific knockout mouse? We are talking about MOTTL3 tissue-specific knockout mouse. For example, let's for now focus on the liver because it's slightly easier. How can we make a tissue-specific knockout of MOTTL3? By using this system. Cree recombinase together with LogSP.
So in one mouse, you put the recombinase downstream of a, for example, liver-specific promoter, and in another mouse, you flag the METTL3 with LogSP, and their offspring will have a liver-specific knockout of METTL3 because the free recombinase don't express in the liver.
Okay, yeah. So that's exactly what happens. So it takes time because you need to breed mice together. But you have 2 mice. Okay, you have 2 mice. One has in its cell, it's a genetically modified mouse, and it has in each of its cells a gene that codes for clay recombinase. But if you are clever, the promoter of this clay recombinase, do you know promoter? What is promoter? Yes? You know?
Sequence that initiator protein, where initiator protein attached.
Yeah. What does it do more generally? There are many proteins who will come to the promoter, but more generally, what does it do? What is the function of the promoter?
The gene expression will be high.
Will be controlled. It can be high, it can be low, depending on the promoter. So the promoter is the region that controls the expression of the adjacent gene. So what you need to have, it's a promoter of a gene that is highly expressed in the liver, for example, but not expressed anywhere else. For example, for the liver, what promoter you could use? I don't know if you know that. What promoter could you use? Think about the liver and the kind of things that are synthesized in the liver. I don't know if you know. No? There is a protein called albumin. It's a very general protein that you find in the body, in the blood. And it's made by the liver. And if you take the promoter of the albumin gene and you put it upstream of the clay gene, and then you transform your mice with this new gene, these mice will only express clay recombinase in the liver because it's controlled by the albumin promoter. That's it. That's only half of the thing. Next, it's your mouse of interest. And you imagine your MOTTL3 gene. These little squares that I am drawing, these are the exons of the MOTTL3 gene. And these are the introns. I should write. This is MOTTL3. This is not a new gene. This is the gene that already exists in the mouse. Okay? This is a new gene. Okay? You make this completely new and you transform the mouse with it. This is the real one, the one that is already inside the cell. But you need to change it. You need to change the sequence of the original gene so that one of the exons of that gene is flanked by LOXP sequence. And I will show LOXP sequence by just a single black dot. Imagine this is the beginning of the gene, so this is the 5' and this is the 3'. This codes for the beginning of the protein, the N-terminal side, and this is the C-terminal side. Where, knowing that you are going to remove with clay recombinase everything that is between two LOXP, where would you put your LOXP sequences in the MOTTL3 gene? So that at the end, you have a non-functional MRTTL3 gene. Where would you put it? Knowing that after you breed, imagine you have a male albumin clay, you have a female MRTTL3 LOXP mouse, and you breed them together. In the babies, the one that have albumin clay and the one that have LOXP MRTTL3, in the liver, the gene will be recombined. But where would you put your LOXP sequence? You want to destroy the gene. You want the gene to do nothing. You want the gene to produce absolutely nothing. Ideal. So where would you put your LOXP sequence? It's very important. If you choose wrongly, you may have all sorts of undesired effects, which I will explain later. Where is the best? You can tell.
What is the effect you want? Because when what do you want? Because I think the baby who receive the METTL3 from one chromosome of METTL3 from the males that don't have the LOXP, it would not have.
And you are right, of course. This is why it takes a horrible long time, because you must get animals who have 2 copies of the MRTTL3 LOXP. If you just breed these mice together, you will have heterozygote mice. And they have a normal wild-type copy of MRTTL3. And this is not good. So you must breed them with other mice who have also MRTTL3. So at the end, you must have MRTTL3 LOXP albumin clay. This is what you want to have at the end. You don't want to have MRTTL3 LOXP wild type, because otherwise nothing will happen. You want both copies of the gene. However, my question was, where it is the LOXP? It doesn't matter whether it's homozygote or heterozygote, that's a different discussion. Where do you want it to be? Where in the gene is a good place to put your LOXP, so that after homologous recombination, after recombination with clay protein, your gene does not make the protein.
Which direction is it reading?
That's a good question. That's the reading direction. So the translational start codon is here, let's say. And the stop codon is here. So where are you going to put your LOXP?
Is the METTL3 located on the X chromosome or located on the memory?
The MRTTL3, it's not, no, it's on, I don't know, maybe the chromosome 11 or something? It's just normal chromosome. Autosome? Non-sex chromosome. Where would you put your LOXP? Where is the best place? I don't remember how many exons MRTTL3 has, probably more than 7, but I mean, it's okay. It can still work that way. Where would you put the LOXP?
Sandwich all the exon.
You are not doing things hard for you. Yeah, you could put it there. You could put it there, but unfortunately, this will not work very well. Depending on the size of the gene, the two LOXP may be too far from each other. So ideally, you want them within 1,000, 2,000 base pair from each other. Not that long. Okay. So, it's a good idea. So, barring this, you cannot remove the whole stuff. Where would be a good place to start?
Start codon?
The start codon. What do you mean the start codon? So let's say the translational start codon, let's say it's here. This codes for the 5' UTR, and the beginning of the protein is really from exon 2. So you would put, tell me stop when you want to put the log speed.
Stop.
And tell me stop when you want to put the next log speed.
Stop.
Exactly. Exactly. If you put the log speed sequence on either side of the exon that has the translational start codon, this is going to make a gene that does not code for anything. You may produce a small peptide or not even anything, because in this case, the translational start codon is completely disappeared. What you might have, however, is you will have transcription. You will have transcription, but transcription will lead to this thing. All the exons may be present, these ones may be present, but this one will not be there because it will be removed. Okay? However, this protein will completely be something that does not exist. And it's likely to be very short anyway, because if you remove one exon, you may have stop codon that appears very quickly, and no protein. However, imagine somehow for whatever reason you cannot put it where you have the start codon. Where would you put it? What is important to consider? So you cannot remove the exon that has the stop codon. You must remove another exon. Which would you choose?
Should reading frame having one?
Yes. Reading frame is very important. The reading frame is very important. Why? Let's imagine that the translational start codon is the first codon of this exon. And this exon is 120 nucleotides. Okay? This first exon, let's magnify. This exon, the ATG is here. This is the translational start. And this is 100, that works, right? 120, sorry. 120 nucleotide. And then you have another exon that is also 120, it's a very regular gene. And then you have another exon that is 121, and then you have another exon that is 119. So that's easy to count. So, and there are other exons. There are other exons that way. You can remove the ATG. You could remove the ATG. But barring the removal of ATG, then the best way will be an exon that is very early in the beginning of the protein. And you have two choices. Either you remove this one by putting a log spin here, or you remove this one, putting a log spin. Which is the best? Why? Yes. Do you understand that?
Frame shift mutation.
Yes. If you remove this thing, nothing will happen because the frame shift is exactly the same. So what you would have is a protein that simply lacks this exon. The exon will be removed, but you will still have the protein. However, if you remove this one, the protein will start from here, will continue, and then it will continue there, but then the codon will be completely different. So the amino acids will be completely different from that point on. And it's very likely that very quickly there will be a stop codon appearing, making a protein that is completely blunted, probably containing only 240 codon plus a few others, and then a stop codon. So this is the best thing you can do. And as you said before, once you make this, for example, here, then you must get 2 copies of the LOXP MOTTL3 in the same time as getting albumin clay into the mouse. Otherwise, what you are going to have is not complete knockout. You are going to have half of the gene, half of the MOTTL3 will be knocked out in the liver, but every single cell will still have the original wild-type copy of MOTTL3. So you must make a mouse that is homozygous for this LOXP mutation that you are doing. At the same time, albumin clay. Is that okay? Is that understood?
I'm just thinking, I mean, generally this is a very good way, but if you already know very well like the structure of the gene like MUTTS, why can't you just remove it, like, why can't you just design a specific way to make the protein unfunctional? Like, I mean, generally, of course, this is a very smart way, but I mean, if you have a very good knowledge of the protein, the gene you are removing, why can't you just design a more specific way to remove the gene?
What do you mean? What do you
for
mean?
example, I don't know. For example, you already know, for example, which axon is the most important. Like it's actually the plays the most important role in the function of MBK-TL. So you just remove that axon. That's what I think.
Yes, you can do that, and people have done that. So, for example, you know all these clock proteins, PER2, for example, and clock. Some people use this kind of reflection, like, for example, obviously this domain of PER2 is very important for its interaction with CRY, for example. So let's remove that domain. But that's not a knockout. Because usually what you will have, if this is the only thing you consider, without considering frame shift and all these things, what you may get is a PER2 protein without only one of its domain. And the effect of that protein may be completely different from the lack of that protein. A protein with one domain that has been removed may act as a dominant negative. So it may be like the knockout, because this protein will have an overall negative function, but it could be a dominant positive as well. You really never know what will happen. Usually the best is completely removing and make sure that a non-functional protein is made. But to know the function of domains, then you have to remove specific exons. That's very true. you are kind of wondering, what is the role of the path domain of PER2 protein? So let's remove the exon that codes for the path domain. So you can do these things.
How do you get the homozygote not spin from MET2?
How do you get? Yes, how do you get? How do you get the homozygote?
You just choose, you just hope that among their structure there will be some male.
Yes, that's right. So the first mouse that you get will be hetero, but you will get many of them. So you just take one hetero male, a few hetero female, you breed them together, and then by PCR, you just analyze the DNA, and you select the pups that are MET3 LOXP.
Why cannot we initially why don't we insert both the CRE adenine CRE with the adenine promoter and MET3 with LOXP at first place?
Yes, so this is usually, you have to choose how you breed your mice, okay? You are right that the best way might be first heterozygote MET3 LOXP plus bred with albumin clay. And then the babies will be MET3 LOXP plus albumin clay. And then you breed them with MET3 LOXP plus. So that the babies, you will have MET3 LOXP albumin clay, and you will have MET3 plus albumin clay that you can use as control. So you have different ways to do, but at the end, you must get LOXP.
Can we start at first, for example, creating a mouse with both adenine CRE and
Both. That will be difficult. In the same time, to do both will be pretty difficult. In the same time, it might be very difficult. However, it becomes increasingly easier to do these kind of things. And usually, these mice, usually you can buy them. There are many clay mice that are available online that you can buy from companies. For DSCN, it's the same, but it's not albumin clay. We are using a receptor gene that is expressed only in DSCN. It's a G protein coupled receptor. And SLC, no, it's a neurotransmitter transporter that is expressed highly in DSCN, and we just do the same thing. But this is an old way. This is a way that many people have been using since 20, 30 years. Now there is a new way, which I taught in the previous lecture, that is much easier. And you can get homozygote knockout in a fraction of the time as doing all this. Do you have any idea? You cannot say because you know. So yeah, you make a mouse, albumin clay. You wait until the mice they grow to the adult age. You make these mice in the same time, and you wait until they grow in the adult age. And then you just turn your thumb until these two reproduce each other, and they make babies, and then you wait, and then you wait, and then eventually you make it. But there is an easier way now, in which only the time it takes to make an albumin clay mice, you can use the same time or even shorter time to make already MET3. You can't, because we are talking about tissue-specific. Forget about what I said. Forget about what I said. Still now, if you want to make a tissue-specific knockout, you must use this. I was thinking of something.
With LOXP, it was originally associated with insects, wasn't it?
LOXP, no, maybe, but I think it's a bacterial thing. Clear recombinase is a bacterial enzyme.
Oh, and what used to, how do they use it? I assume they don't want it to be neural genes.
I don't think it's a bacterial. I think it's a bacteria. Maybe the first time it was used was in Drosophila, as usually genetics experiments are. But I will check when the first time the Cray LOXP was used. Well, I think the Cray recombinase and LOXP, these are really bacterial sequences.
How does the bacteria use it? What do they use it for?
That's a good question. What could they use it for? What could they use it for?
Would it be to fight off viruses? But then you would have to put lots of sequences.
Yeah, it could be linked to antiviral defense, or it could be linked for just to be able to remodel your genome pretty quickly and to adapt to different conditions. Like the new method I was thinking about was the CRISPR-Cas9 system, and CRISPR-Cas9 system is a defense against. It's an intracellular immune defense. But Cray LOXP originally, how it was discovered and used, I need to check. I don't know this one by now. But still now, if you want to do these tissue-specific knockout, you have to pass by these breeding animals that have Cray recombinase and that have sequences. Okay. So, now that it's okay, it takes time. This is what we are doing now. But for this current investigation, you are doing in vitro stuff. You don't have mouse yet, and you are trying to convince people that RNA methylation is important for the clock. Thinking about what we have seen before, especially things like what we showed in the figure here, that the inhibition of M6A by diazadenosine was accompanied by a decrease in RNA processing. So, thinking about that, what would you do after demonstrating that only decreasing MTTF3 protein is enough to cause elongation of the period? So what could you do next?
Isn't it enough?
Hmm?
Isn't it enough?
No, it's not enough. No, that's the problem, you see. It's not enough because we did not show MTTF3 knockout mice. The reviewers, the people who are reading this paper, they need to be convinced hard, especially when you don't have animals to demonstrate that what you're doing is specific and everything, then you need to do a lot of things. It's not enough. It's not enough. And at that time, the animals were not ready.
What aspect of the circadian clock is controlling?
See?
Which genes in the circadian clock?
Yeah, so you could, that's true. You could, for example, here obviously PER2 is affected, but it's the period, so you don't know if PER2 is directly affected or not. So you could certainly check which clock gene is affected by the knockdown of MTTF3. And I think if I remember well, we did something like this. We did something like this here. So I have shown that to you already. So the first thing we did is the same experiment as we did with DAA. if we knock down MTTL3, we should see a decrease in the processing of RNA, just like what we saw by using DAA. And you can see this figure is almost the same as the previous figure with DAA, but in this case, when we do the MTTL3 knockdown, we have an obvious decrease in RNA processing, just like this figure here earlier in the paper, just like this one here. It's very similar. So this again shows that DAA and MTTL3 knockdown, they are very similar. And then what we also did is this one here, showing that what we think is happening is that we are sort of making RNA processing slower because we are inhibiting RNA methylation. So what we decided to do is to check two clock genes, BMR1, this is the name of the gene, but this BMR1 is more often used, and PER2, and we quantified for 2 days the amount of pre-mRNA, which is this line here, and the amount of mRNA in the nucleus, which is the interrupted line, and in the cytoplasm. So we can check different steps. We can check pre-mRNA, which is transcription. We can check nuclear messenger RNA, which is splicing. And we can check cytoplasmic messenger RNA, which is nuclear export. So we are trying to get an idea of what happens at every step of the way. And we do that under normal cell, under normal condition, and with MTTL3 knockout cells, knockdown cells. And what we see, especially when we see BMR1, is that every single step is actually elongated when you knock down MTTL3. So for example, here, if you check the delay in the peak of mRNA, of pre-mRNA, compared to the peak of messenger RNA in the cytoplasm or the nucleus, you have about 4 hours difference here, peak to peak. But when you do MTTL3 knockdown, the difference between the pre-mRNA peak and the messenger RNA peak becomes 8 hours. So there is obviously a delay that occurs. And if you check the difference between the trough of the nuclear and cytoplasmic messenger RNA, the trough is the same in control cells, but there is a 4-hour delay between the nuclear mRNA and the cytoplasmic RNA and the MTTL3 knockdown. And the same thing, but a little bit more subdued, is happening with PER2. So indeed, it seems that by knocking down MTTL3, we have made the RNA processing slower, especially for BMR1. But still, it's not enough. It's not enough. Because it's again in vitro. So what could we do to sort of say, hey, RNA processing is very important for the clock. So far, we have inhibited M6A, which we know it's important for RNA processing. So what? So what? And maybe you're right, maybe the last figure might not have been necessary. I don't think I submitted it without the last figure. But I thought the last figure might sort of make it a bit more interesting, so I submitted. But maybe the last figure was not necessary. So what else could you do? This is not the last figure, this is one before the last, I think. Isn't it? Right? Okay. So what else could you do? Here we show that M6A dependent RNA processing is important for the clock. So what we thought of doing, then if M6A dependent RNA processing, we know that M6A is important for nuclear export. Okay. So maybe if we directly inhibit nuclear export, we will have the same effect. We don't inhibit M6A, but we inhibit export of messenger RNA. So this should be the same. We should still see a period elongation. And this is what we decided doing. In this case, it's a little bit maybe more difficult to explain, but what we did is, as you can see, this is very similar to the previous experiment, but we knocked down two different genes. Here, they are not related to M6A, they are related to RNA processing. RNMT and NCBP1. What are these guys? What are these guys? RNMT is a protein that is linked to the cap. It's a protein that recognizes the cap and methylates the cap. Because methylation of the cap is known to also be important for the export of messenger RNA out of the nucleus. So it stands to reason that if you knock down RNMT, you will also have an elongation of the circadian period. And you will also observe a decrease in RNA processing. So we did that. We knocked down RNMT. We don't see any almost no RNMT left. And if you check RNMT knockdown, we have the same as what we observed with DAA. So the nucleus is still labeled with messenger RNA after 4 hours, even though in the control nothing is seen. And when we knock down RNMT, we have an elongation of the circadian period. Same stuff. Then NCBP1. NCBP1, it's a protein that recognizes methylated cap and takes the mRNA out of the nucleus. It's a protein that interacts with RNA. So therefore, it stands again to reason that if we knock down NCBP1, we should see the same effect. And we have elongation of the circadian period when we do NCBP1 knockdown. And we also have a decrease in the disappearance of the messenger RNA degradation. And then that was the last figure, which we conclusively demonstrate that RNA processing is important for the circadian clock in vitro. And M6A, inhibition of M6A by decreasing processing of messenger RNA is also important for the biological clock. So that's it.
Is nuclear export important? And previous figure, we saw the difference between pre-mRNA and nuclear mRNA is significant. So
Yes, exactly. So the time it takes for splicing may also be important. The time it takes for splicing, how much time the cell takes to splice messenger RNA might be important. If splicing is slower, then maybe the clock will also elongate. In this case, we have not shown any specific mechanisms. It suggests that RNA export and splicing is important. But we don't know which mechanism. The very big problem with RNA is that everything is linked together. The problem is in textbook, like for example, molecular biology of the cell or essential cell biology, the book that I'm using for the other lecture. Yeah, I will stop this one now because it's the end. So I just keep. one of the most researched subjects at the moment in the clock and other area of biology is that what you have in textbook is you have DNA, then you have pre-mRNA, then you have major RNA, then you have nuclear export, and so on and so forth. It's all very stepwise. 1, 2, 3, 4. But actually, this is not like this. Of course, nuclear export is different, but all these things here, they are timed. They occur at the same time. So if you inhibit splicing of RNA, you will also decrease the speed of transcription. If you decrease the speed of transcription, you will also decrease the speed of splicing. They are all tied together, and it's sometimes very difficult. Most of the time it's difficult to separate things into separate steps. So this is something that people are heavily doing now, trying to know exactly which process is affected. But they co-regulate. And then something that has been shown recently about RNA methylation is that this is really crazy. M6A regulates the speed of transcription. M6A regulates the speed of transcription, but this also affects translation. This is crazy. So the amount of if the transcription is very fast, the amount of M6A is low because the cell does not have much time to methylate messenger RNA. So if transcription is very fast, then the RNA will not be so heavily methylated, and translation will also be low. But if transcription is very slow, then it will have which direction it goes. So it's really kind of a recent paper, and it shows that the speed of transcription affects how much RNA methylation there is, and how much RNA methylation there is affects translation, which means the transcription rate affects translation rate, which is completely separated process. Translation happens in the cytoplasm, and transcription happens in the nucleus. But there is something happening at the level of transcription that then determines the rate of translation. So these kind of things show that there is really everything is linked. So I think it's yeah. So in the paper, we have not really shown which process affected by M6A is the most important for the clone. We haven't shown that. We just suggested that it might be RNA export, it might be splicing, but we don't know which is the most important. There is not this message. Any comment, question? I will not go into that today, but there are many other data in the supplementary figures of this paper. And we also show that nuclear export may be affected, splicing might be affected, but also RNA degradation might be affected. When we inhibit RNA methylation, the stability of messenger RNA seems to be high, which may also contribute to the elongation. So there are many stuff. M6A really impacts on everything in the in the lifetime of the messenger RNAs. So, anyway, that's finished for today. So if you have a question.
Is coronavirus plus on Saturday?
No! God, no. God, no, it's not. There is no Saturday, right? Saturday was only 2 Saturdays, right? More than. Yes.